Sargis, do you want me to give you a countdown?
Oh, I was waiting for the countdown. Are we on air? Okay.
Yeah,
Sorry.
so sorry. Yes, sorry about that. I had thought you were going to minimize and then jump back in, but I will give you a countdown now. Okay,
Okay, sounds good.
Starting in 5, 4, 3, 2.
Hi, my name is Sargis Sedrakyan. I'm a junior faculty at the University of Southern California and Children's Hospital Los Angeles. I thank the organizers for this invitation and the opportunity to present our work. Today, I will talk about extracellular vesicles and chronic kidney disease. So what is chronic kidney disease? It is defined by gradual loss of renal function over a period of time. Sometimes it's short, sometimes it's relatively long. And there are many different factors that contribute to the initiation progression of chronic kidney disease. And in the population, the two main ones being diabetes and cardiovascular problems. In addition, obesity, genetic mutations, tobacco use, and aging are also important factors that can lead to progression of chronic kidney disease. I will not exaggerate if I say that kidney is one of the most complex organs that we know of, and as it's very complicated and heterogeneous in structure.
Hi, Sargis, can you hear me? Sargis, can you hear me? If you can hear me, you've frozen. If you want to try exiting the studio and re-entering.
Am I back?
Yes, there you are.
Yeah, sorry, I didn't realize. Where did you lose me exactly?
I believe it was towards the end of slide 2, so we could just do a fresh restart if you want.
Um, Up to you. I can start from slide 2, or
Yes,
you tell me exactly.
Let's go ahead and start since you were on slide 2. We're close enough if you're okay with that, and then we can give it another shot. And then if anything comes up again, I'll let you know.
Yeah, so what happened on my end was that my slides started changing without my touch. I didn't see your message, actually. First, I saw my slides going haywire. Then I realized I'm not, something is wrong.
Yeah, I figured you weren't able to hear me, so I thought maybe one way to get your attention was to move the slides around. So
I see. Okay, that's a good way.
Yeah.
Okay.
So I'm glad you picked up on that. That's good.
Okay, well, that's a good way to wave the flag. Okay.
Perfect. Maybe what could have caused the potential interruption? Well, you did click through all your slides, correct?
In the beginning, yes, I went through all of them.
Okay, good. Just want to make sure that we kind of remove anything else that could cause interruption. So I think we're good to go, and we can give it another shot. So I'll give you a countdown, and then you can start again.
Oh, from slide 1?
Yeah, if that's okay.
Yeah, that's fine. As long as I know where I need to, kick in. Sure.
Okay. All right.
I'll wait for the countdown. Yes, thank you.
Definitely. Okay, starting in 5, 4, 3, 2.
Hi, my name is Sargis Sedrakyan. I'm a junior faculty at the University of Southern California and Children's Hospital Los Angeles. And I thank the organizers for this invitation and the opportunity to present our work. Today, I'll talk about extracellular vesicles and chronic kidney disease. So what is chronic kidney disease? It is a kidney disease defined by gradual loss of renal function over sometimes extended period of time. It can be years, and many years. And there are many different factors that contribute to the initiation and progression of chronic kidney disease in human population. The two major ones being diabetes and cardiovascular problems. In addition, obesity, genetic mutations, tobacco use, aging, and other factors as well do contribute to progression of chronic kidney disease. I will not be exaggerating if I say that kidney is one of the most complex organs in the human body, being that it is very heterogeneous in nature, structure-wise, and very difficult to study. So the functional unit of the kidney is the nephron, which is composed of the glomerulus and tubules. The glomerulus is a bolus structure that contains microcapillaries made up of endothelial cells and podocytes, as shown in the diagrams in the right. The podocyte is the cell that's highlighted in green that surrounds the capillary structures, and between the endothelial cells and podocyte lies the glomerular basement membrane. And in the electron micrographs on the bottom, you're seeing a projection of the glomerulus in a scanning electron microgram actually on the left. And on the right is a close-up image where we can more clearly see the basement membrane. It's a thin layer of matrix. And on both sides, we have fenestrated endothelial cells and the podocyte. What's so special about this structure is that nephron cannot be regenerated in adult life. So we humans are born with specific numbers of nephrons, and they are predetermined. Usually that number is roughly somewhere around between 500,000 to 1 million per kidney. The glomerular cells, namely the podocytes and endothelial cells, are very unique in their nature. The podocyte is unique because despite the fact that it's a typical endothelial cell, its location, architecture, its function are unique because they have these food processes that make interdigitations at the filtration barrier that determine the perm selectivity nature of the filtration function. The podocytes are post-mitotic cells. That means they do not proliferate. They cannot be replaced. And if they do so, that means there is a problem. And usually this happens during disease. They slough off from the basement membrane, which usually is associated with onset of proteinuria. Glomerular endothelial cells are unique as well because they have fenestrations and they are responsible for the first filtration activity, as the ultrafiltrate from the capillary first has to leave the endothelium, then the basement membrane, then through the podocyte. And lastly, the glomerular basement membrane is unique. And I want to highlight this because this matrix is important and plays an important function in the filtration. It's composed of collagen type 4. This special collagen that is produced only by the podocyte, and mutations in this collagen or any other component of the matrix can lead to disease, and I'll talk more about this later. So the homeostatic balance between the glomerular cells and the glomerular basement membrane are important for the maintenance of the ultrastructural integrity and function of this filtration unit, and any alteration of any of these components can lead to chronic kidney disease. So the main treatments for kidney disease are hemodialysis and kidney transplantation, and we know that loss of glomerular function leads to development of Tyler. Hello, Tyler.
Yes.
Can you hear me? Yes, I'm having a problem when I switch the slides, right? Half of my slide disappears from my screen, and so I'm kind of misled. I don't see everything on my screen. What should I do? Every time I click, should I click the next, or because I'm using the keys on my keyboard? Every time I click, my slide just slides up on my screen, and I don't see half of it.
Okay, could be the keypad that's causing a little bit of a glitch. So if you want to try using the next and previous and see if that works okay for you.
Sure. Can I repeat this last slide because I got tracked off because of that?
Yeah, this is slide 4, correct?
Yes, slide 4.
Okay, let me just make a quick note for it.
Mm-hmm.
Okay. I've made a note, so whenever you're ready, just give it a few seconds and then you can start again.
How do I know when to start, or it doesn't matter? You just tell me the...
Yeah, just wait a few seconds of silence, and then you can pick back up.
Okay, sounds good. So loss of glomerular function leads to development of chronic kidney disease. And in the United States alone, there are more than 30 million patients, Americans, who have the diagnosis for chronic kidney disease. And this is a major burden financially on the healthcare system. And in 2018 alone, the estimated cost for CKD treatment and end-stage renal disease treatment was somewhere around 114 billion. So what are some of the treatments? We are not very fortunate because the only treatments currently available for patients with renal failure is either dialysis or kidney transplantation. So and the projections are not looking very good. If we look in this graph, we can see that between 1999 and 2015, the number of transplantations were relatively constant, somewhere around between 17 to 18,000 annually, whereas the number of patients in the transplant waiting list, waiting for a donor organ to have a transplantation, has been growing over the years, and these numbers are even higher, currently. So this is a tabulation of all the different, etiologies or kidney diseases that lead to eventually kidney transplantation. As you can see, the number is huge, and the conditions range from renal cancer to familial conditions to glomerular sclerosis, and even sickle cell anemia can lead to kidney disease. So the conditions are enormous. If we take a historical look as to where we stand right now and what are the opportunities, as I mentioned, dialysis and kidney transplantations are two mainstream therapies for kidney replacement. But both of these were discovered back in the 1940s and 1950s, and only in 1983 we had the introduction of immunosuppressants, which really enhanced the success of kidney transplantations, providing support for the against rejection of the transplants. So in addition, treatments for CKD, in addition to dialysis and transplantation, have been centered on slowing down its progression by systemic approaches, such as using diuretics or ACE inhibitors, which have many side effects and are not specifically targeting the glomerulus. So we're in 2020 now, and no new developments in terms of approved therapies are known. And so some of the areas that the research is working on is gene therapy, tissue engineering, and regenerative medicine applications, and even some pharmacotherapy approaches to find a new therapy. In addition, there is the area of stem cells and extracellular vesicles that have gained a lot of traction in the recent years, and there seems to be some opportunity here for finding a new possible therapy. And so our question is, can we apply stem cells or stem cell-based products to target kidney disease or renal complications? And so over the years, we have took a specific approach of using stem cells for potential therapeutic to treat kidney disease. And in the past, we have looked into using AFSCs for treatment of acute kidney disease, as well as chronic kidney disease. And we have successfully shown that single injection of amniotic fluid stem cells provides renal protection in terms of morphology and function, as well as survival in these animals. And more recently, we have taken a new approach where we isolated extracellular vesicles from amniotic fluid stem cells, and we've shown that just like the AFSCs, they provide similar protection. And in particular, we observed that these EVs were able to protect endothelial damage induced by VEGF signaling in Alport animals, and I'll say a little bit more about this in the next 2 slides. So just like any other cell, AFSCs do produce a lot of extracellular vesicles, and we isolated them using ultracentrifugation and characterized them by nanosite. And we can say that AFSCs produce large quantities of EVs so that they can be injected in large number of animals. So by nanosite, we characterize their size to be roughly about 200 nanometers in diameter. So these EVs express typical mesenchymal markers, extracellular markers such as CD9, CD63, as well as angiogenic markers such as VEGF receptor 1, receptor 2. So these 2 receptors are important for later points that I will make in the next slides. In addition, these vesicles also have a large bank of microRNA, both pro-angiogenic and anti-angiogenic, and obviously anti-angiogenic microRNA were detected to be much higher, and this was important as well. So moving forward, I'll introduce the animal model we used, which is Alport syndrome, to study CKD. This is a genetic familial disease that has mutations in the collagen 4, alpha 3, 4, 5 chains. In our case, our model has a mutation in alpha 5 chain, which results in abnormal deposition of the glomerular basement membrane, leading to progressive loss of renal function and ultimately kidney failure. In this image, you are seeing again an electron micrograph of the basement membrane in histology showing normal morphology of the kidney. And upon the mutation in the collagen 4 alpha 5 gene, we have a severe disruption in the basement membrane, as you can see in the projections in the right, and development of fibrosis in the glomerular and tubular compartments shown by this histology. So this leads to infiltration of immune cells, deposition of extracellular matrix, scarring, and reduced blood flow. And the reason we're using this specific mutation is because 85% of human cases have this specific alpha 5 mutation. So it's a very common, whereas the other mutations are much more rare. So in addition, we made this interesting observation that VEGF, which is very important for the maintenance of the glomerular integrity and specifically the endothelium and the fenestrations, is highly elevated early on in Alport mice in the glomeruli. And so we were very interested to understand whether the vesicles, which express now VEGF receptors, could mitigate this. And so we performed some in vitro experiments where we cultured endothelial cells from glomeruli, and we treated them with VEGF just to recapitulate, to mimic the conditions that exist in the Alport glomeruli, and treated them with normal EVs and EVs that were knocked out for VEGF receptor 1. And interestingly, we observed that the normal EVs were able to capture or bind to VEGF in the media and prevent this VEGF from being taken up by the cells. And subsequently, we performed some further analysis looking into the cells and did some immunoprecipitation assays where we confirmed that there's a direct interaction between VEGF and the receptors expressed on the surface of the EVs. So this was important, but then it was also important for us to understand whether the EVs, once injected in mice, could really home to the kidney. So in collaboration with Dr. Bisolati in the University of Torino, we performed these experiments where the EVs were injected in mice, and we could easily detect them in the kidneys and image them by bioluminescence, as shown in this figure. Next, obviously, was also important to understand whether they have any therapeutic potential, and so we looked at their renal function in terms of creatinine and albuminuria. And interestingly, with one single injection, we could see significant improvement in both of these parameters. So moving forward, our ultimate goal was eventually to be able to translate this technology. And so our next steps as we move forward, we switched from using old mouse system where we had the mouse model and using mouse EVs to a mouse and human hybrid model where we now still use the same mouse Alport syndrome model, but apply a human IFSC-derived extracellular vesicles to understand their biodistribution, therapeutic effect, and mechanisms of action, as well as understand mechanisms of disease progression in these animals. So in this, for this aim, we developed certain directions, namely 3 specific directions that we aimed our studies on. In the one direction, we developed key assays to allow for consistent characterization of these human EVs now derived from consented donors and evaluate their potential for clinical translation or application. In the second direction, we really thought about the possibility of expanding these EVs to clinical scale. So we employed GMP-compatible 3D hollow fiber bioreactor to be able to meet this need. And then finally, we are also developing a non-invasive, clinically applicable in vivo tracking system for EVs using MRI to evaluate potential homing and associated toxicities in undesired off-target organs or tissues. That will eventually, combination of these three approaches will allow successful translation. So looking into the key assays, there are three of them. The first one is looking at their identity and purity, size distribution, production and characterization by their cargo, looking at the protein and microRNA. Second assay is the potency assay in vitro, where we look at the functionality of the EVs, specifically looking at the capability of the EVs to trap VEGF, as I showed earlier. And then lastly, look at their therapeutic efficacy in vivo, where we isolate glomeruli in injected mice and look at VEGF modulation, analyze gene regulation by RNAseq, and of course, functional changes. So looking at the first assay, where we do the identity and purity, we characterize the EVs by nanosite, just like the mouse ones, and we determine their size and distribution and concentration. We also employed ExoView technology, where we're able to visualize the EVs based on their expression of tetraspanins, the CD9, 63, and 81. And we can really, this technology allows is a combination of flow cytometry and immunohistochemistry that, as I said, allows visualization. As shown in the right, we can see really single EVs and identify the expression of specific markers in addition to tetraspanin and really understand their heterogeneity. So this allows us to easily characterize them. So in addition, we looked at surface markers. As I said, the cells are coming from consented donors. So in this example, we're looking at two samples, clonally derived extracellular vesicles from two consented donors compared to the mesenchymal stem cells. And as you can appreciate, these two independent consented lines have very similar expression profile. So next, we looked at their protein cargo and microRNA, obviously by RNAseq and mass spec. And in this diagram, you are seeing the 10 most highly expressed proteins and microRNA. I should say that we identified 675 intact proteins in this glomeruli, and 2,535 microRNA were expressed. And this profiles really allow us to put a fingerprint or on the EVs that characterizes or puts an identity tag that we're able to kind of identify and characterize them each time we have a new line. From our experience, using the culture methods and isolation methods and characterization methods, we're able to get consistent lots of human EVs yielding the same features as highlighted. So next thing I want to point out is that when we compare our data from the human EVs and mouse EVs, there is an 80% overlap in terms of protein expression, which confirms the validity of our derivation method. From the proteome analysis, we in the human sample, we also confirmed the absence of HLA, DR, CD80, and CD86. So this is important in terms of immunogenicity. And sequencing, RNA sequencing also confirmed that the human EVs do not carry the major microRNA involved in adaptive and innate immune responses, such as MIR24, 155. So this is important as we move forward using the human EVs. So our assay number 2 is the potency assay in vitro, which I talked about for the mice. And so the same was applied here, and the endothelial cells, glomerular endothelial cells, were cultured and treated with VEGF to mimic the condition that we have in outward glomeruli. And then the human EVs were applied and very similar to what the mouse EVs did, we saw amelioration of VEGF protein or decrease of VEGF protein in this assay. And in the follow-up or subsequent experiment, we performed the same assay without the cells, without the confounding factor of cells. And again, we observed a direct interaction between the VEGF and the human EVs, confirming the potency of the human-derived EVs being similar, if not better, than the mouse. So this was very encouraging. And then lastly, looking at the in vivo efficacy of the human-derived EVs in this experiment, 2.8 times to the 10 power EVs were injected into the 2.5 months age-old Alport mice. And as you can see, the single injection significantly improved VEGF activity or activation in isolated glomeruli in this example. And lastly, we also measured the proteinuria in these animals. And again, injection of the human EVs significantly ameliorated proteinuria 10 weeks time period after the injection. So moving forward, I'll give you a little bit information about the RNA-seq analysis we performed. So mice that were injected with the EVs and mice that were not injected, we isolated their glomeruli 1 week after the injection and sent for RNA sequencing. And this is the data that is being projected in this PCA plot. As you can see, the wild-type samples on the left, the diseased Alport mice with the green are, separating very nicely in this 2D plot, whereas the injected samples shown in blue are really separating from both, and they are actually improving at least in one dimension on the y-axis. So this is very encouraging. We're seeing some effect. We performed a lot of comparative analysis between all the groups and to understand the differences and similarities and how the injection of EVs is really changing or shifting the pathways in the glomerular cells. And so very briefly, looking at some pathway enrichment analysis, we identified the key mechanisms key mechanisms or pathways that are affected in Alport glomeruli compared to normal. And also looking at the injected animals, we now understand some of the mechanisms that might be involved in leading to improvement in renal function. And as we look further, here I'm showing just two examples in the lipid metabolism panel and cell matrix adhesion molecules, and shown in red with red dots, you can see the number of genes that are being modulated or reversed to normal in this glomeruli. So, in addition to lipid metabolism and matrix adhesion, there are some other areas where the EVs are actively, having a positive effect, and we are, continuously studying these mechanisms. So next, I will talk about the 3D hollow fiber bioreactor studies that we are conducting. So we know that EV clinical translation is limited by scale up, as I said, and hollow fiber bioreactor can support culture of large number of cells at high densities and can produce large number of EVs. And so in collaboration with fiber cell systems, we are culturing the human consented AFSCs. And as you can see in this figure, I apologize for the low resolution, but the cartridge that's shown in the front is loaded with AFSCs and fed by media, and without having to passage the cells over a very long period of time, we can continuously harvest large number of EVs. And so amniotic fluid stem cell EVs grown in this cartridge produce biochemically and functionally equivalent EVs similar to the 2D derived EVs that we growing in our culture room, with yields that can support clinical scale and GMP compatible system. And so just like with the 2D EVs growing in our lab. We performed the same assays for the bioreactor derived EVs, and as you can see in this figures, the potency assay was not any less than the 2D derived, was even better, and the improvement in proteinuria was much more better than with the 2D system. So this is very encouraging, and we're looking forward to improving this even further. So next, I will talk about the next the direction or next area we are advancing or working towards is the MRI detection of EVs using special dyes, or more specifically iron particles, to study biodistribution and potential homing and off-target associated toxicities and etc. And so MRI is an important modality, but before I go to the MRI, I should mention that there are other modalities, imaging modalities currently available, such as optical imaging through bioluminescence and fluorescence imaging, nuclear imaging, that can potentially be used for tracking EVs in vivo, but they are, both of these have certain limitations, mainly due to the safety concerns, because one involves radioisotopes, and optical imaging requires transfections, plasmids, and fluorescent dyes. And so, in contrast, magnetic imaging or MRI is much safer because it uses only iron particles. And provides excellent spatial resolution that can be used to identify where the EVs are. So in collaboration with Visicell Medical, and that specializes in MRI, cell, and EV tracking technologies, we're testing the possibility of using MRI to study EV biodistribution in vivo. So we performed a proof of concept studies demonstrating that VMI labeling agent is safe on cells and EVs production and does not affect their viability, as shown in the first panel. That the labeled cells or EVs can be detected with electron microscope, as shown by the electron dense projections in panel C. And importantly, this labeling agent does not affect the size distribution and production levels of EVs, as we confirmed with nanocyte tracking. And importantly, we can use relaxometry to measure the iron content in these vesicles. And importantly, the labeled EVs can also be visualized with MRI. So to test the in vivo tracking potential of the VMI dye, we injected healthy and controlled mice with the EVs with a single dose via two different routes. One was the retroorbital route, and intracardiac route was applied. Mice were imaged once before the injection and 30 minutes post-injection. And as you can appreciate from these projections, a positive signal was detected only in the Alport mice injected via the intracardiac route. That is nicely shown in the blow-up projections of the renal cortices in the right. Our MRI findings were further confirmed by tissue histology, and as you can appreciate in this micrographs, Prussian blue staining is observed only in the kidney injected with the endocardiac route and not through the retroorbital route, and it's only present in the diseased tissue and not in the control mice. So intracardiac delivery of EVs to the damaged kidney is more efficient than IV route, and we know that the disease really contributes to the homing of the EVs or cells to the target organ. And so this is an important step moving forward, as understanding the biodistribution and homing of any reagent that's being injected into a human organism is important and needs to be well documented. So with this, I would like to conclude by stating that our protocol for human EVs derivation is reproducible and allows for derivation of a large number of EV lots with similar identity and purity and potency. That human-derived EVs show strong in vivo efficacy, as shown by our assays, and that the production of large number of EVs using the 3D hollow fiber bioreactor system is similar to, if not better, than the 2D derived human EVs from our culture, laboratory cultures, and that the combination of MRI technology and the VMI tracking system is highly applicable technology for real-time in vivo tracking of EVs that can ultimately shift the paradigm in safety. And so with this, I would like to conclude and thank everybody in our research group for their individual contributions. I would like to thank our contributors for their support and with the experiments, with their ideas and advice, and ultimately to all our funding agencies for their support. Thank you. I'll take any questions.
Okay, awesome job. Well, we're all set. I will do some quick editing on the front and back ends, as well as just the short slide redo when we have the slide hiccup, and then we'll be ready for the event next week.
Sounds good. Was I within the 30 minutes? I didn't have any timer to follow, actually.
No, you were. Just because we did the restart at the beginning there, I know the time was longer than what it actually was. So you're right around the 30-minute mark.
Oh, awesome. Great.
Perfect.
Excellent.
Any questions for me before we wrap up here?
Not really. So the event is on the 23rd. And this will be available online thereafter, right, for some time?
Yeah, so the live event is on the 23rd next week, and then the event will be available on demand for 6 months after.
Oh, 6 months after. Okay. Sounds good. Well, let me know if anything went wrong and we need to go over it again. I mean, any slides, any section that doesn't look good.
Okay, that sounds good. Yeah, I'll definitely reach out if anything does come up. And then if you have any questions at all between now and the time of the event, feel free to reach out to me.
Perfect. Thank you, Tyler.
Thank you so much. Have a good rest of your day.
Likewise. Bye-bye.
Bye.